Good afternoon and welcome to our October edition of our Sidewalks to Science webinar series. Due to a scheduling conflict, this webinar was previously recorded. We are excited to welcome Komen scholar Dr. Yibin Kang as our presenter. Dr. Kang is a Warner Lambert Park Davis Professor of Molecular Biology at Princeton University. His research is focused on a sticky protein called E-selectin that is found in blood vessels and is thought to promote metastasis. Dr. Kang and his team are working to determine if blocking E-selectin with drugs reduces the ability of breast cancer cells to spread. Dr. Kang received his first major national level grant from Komen in 2005, helping launch his renowned research career. For the past 12 years, Dr. Kang's lab has been funded by Komen, including 4 postdoctoral fellows, 2 of which are currently funded. He is also part of Komen's new crowdfunding initiative, BeMoreThanPink.org, that enables supporters to donate directly to 4 individual Komen research grants focused on metastatic breast cancer. All donations received for the grants will go further thanks to a matching program from our friends at Odinate Therapeutics. The link to the site will be available after Dr. Kang's presentation. Dr. Kang welcomes any questions you may have and will gladly answer them via email after the viewing of this webinar today. If you have any questions today for Dr. Kang or a Komen staff member, please click on the Q&A button or chat on your panel and enter your question along with an email address. Please welcome Dr. Kang.
As Michelle mentioned, my career has been so much linked with Komen, so every time I come to Dallas for the Komen event, it's always like homecoming to me. So I'm going to also tell you a little bit of my story. And you see my picture when I was a little boy, just like Dr. Jensen. So my hometown is actually not too far away from Dr. Jensen, Laos. So my hometown is in, as you can see, if I can use the, is there a laser here? No, don't worry about it. So my hometown is in the southeast coast of China, and I was born in a small fishing village, and I grew up in a small town. And so this is when I was maybe 15 years old. I knew I want to be a scientist, and so this was me in the left side, and with my friends interested doing some little science experiment with plants. So when you ask me about my journey to become who I am today, it really is literally a long journey. So as you can see, I grew up in southern China, and then went to high school in Beijing, college in Shanghai, and then traveled to the US when I was 21 years old. Studied in Michigan and Duke University for my graduate training, and then went to Memorial Sloan Kettering to train with Dr. Joel Massaguet before I joined the faculty of Princeton in 2004. So here I was, maybe 2009, 2010, and I was still interested doing science and surrounded by young people as before. So you can see this is a long journey of more than 30 years. And the reason why I'm doing breast cancer research is actually a little bit different from what motivated Dr. Jensen to study breast cancer in the first place. It's not because it was started with a personal reason, simply because it attracted me as a tough scientific question. When I was a graduate student at Duke, I studied retrovirus, and one of the most dangerous and most famous retrovirus is HIV. So this is the disease that could be very severely affected by retrovirus, but actually most viruses do not actually cause a lot of very deadly disease among humans. But it's a great tool to study how viruses interact with the cells, because viruses have a very tiny genome. They only contain as few as 3 or 4 or up to 9 genes, compared to tens of thousands of genes that we have in our genome. But it's a great, you wonder how such a tiny virus could cause such a tremendous disease like HIV. So this is what I did in graduate school at Duke, is figuring out how the virus is so smart to utilize some of the machineries of our host cells to replicate and cause many of the disease, such as the immune deficiency. But nowadays, we actually know a lot about the viral application, and we have medicines that will control disease even as dangerous as HIV. So when I finished my graduate training at Duke, and I was looking for the next big question to tackle, and breast cancer came to my mind, because as we all know, this is a disease that affects many millions of women worldwide. Almost, many families are affected one way or the other. And different from virus, this is the enemy from within. It's our own cell through mutations becoming our own greatest enemy. And they also, just like virus, they take advantage of the host tissue to metastasize, to spread to different organs. And this causes a lot of trouble. 90% of early stage breast cancer will have recurrence as metastasis. And almost all metastatic breast cancer patients, many of them will die from disease. And we have up to 40,000 still dying from disease every day. These are the statistics that we are all familiar with. But when I start looking at this question 16, 17 years ago, in 2000, when I joined Dr. Joan Messinger's lab for my postdoctoral training, you look around in the field, it's like a big elephant in the room. Everyone knows this is the biggest problem in terms of how patients die from breast cancer, but there's very little effort put into it. Not because it's not important, but simply because it's too hard and we have very little tool to study that. So this is how I get started. I want to study it because I feel this is the biggest question that I want to tackle. But along the way, I met people like you, a lot of the patients, my students' mother. That changed how I think about what I do in the lab. It's no longer a purely scientific interest. It really is a personal responsibility to come up with an answer for people that they consider you as the last option to come up with when they have metastatic breast cancer. So my career as an independent scientist in the last 13 years has been completely dedicated to study breast cancer metastasis. And I was fortunate to work with a number of very talented people. So we've published over 140 papers in the field. And I was actually the person who get the fastest promotion from assistant professor to endowed chair professor in a time frame of 8 years. And now, so among the many titles and honors I received, I think the one that I feel most privileged to receive and most proud of is the last one that you see in here. That was the recipient of the AACR Outstanding Investigator Award in Breast Cancer Research, and this is funded by Susan G. Komen. So this was me receiving the award at San Antonio Breast Cancer Symposium. So you might think that my whole career is all rainbows and unicorns, but it seems such a smooth sailing, but it's actually not true. People in my lab will tell you how much frustrations and how much failures we have to endure to come to where we are today. So as a young scientist, when you started out and you are given a job offer to start a lab at Princeton, and you are all excited, and then the reality starts to set in. You move to Princeton and you walk into your lab, and this is what it looks like, right? It is shiny but it's empty. It's only you, and you have to start from somewhere. As you can imagine, when you try to move something, the hardest part is at the beginning. You need to get it start moving and give it momentum. That is the most important thing. So where do I get the momentum? So this is the list of the supports that I have received from Komen. My first independent grant, national level independent grant as an independent scientist, is a Komen independent investigator grant that get us studying how certain cancers are high risk and likely to metastasize. So that's back in 2005. The next year after I started in Princeton. And then in the middle, I was continually funded by Komen to study bone metastasis. And then last year, I was selected as a Komen scholar and received a Komen Leadership Award, continued to study bone metastasis. Not only me, at least 4 of my fellows, postdoctoral fellows, have received Komen postdoctoral fellowships. 2 of them already become professors. One already actually have become a full professor and associate director of the institute. One just became a professor, and then the third one is about to become a professor. And the fourth one just starting this month as a Komen scholar, Komen fellow. So as you can see, my lab is non-stop, continuous funded by Komen in the past 12 years. And so it's Komen that get my lab started and actually continue to give us momentum to keep moving forward. So I'm going to briefly go over some of the Komen funded research that we have done in the lab, and you can see the potential impact of that could have on the patients and how that could improve the treatment of breast cancer. So you will see I use a lot of the covers. We got fortunate to make some of our research was made as covers, and those are supposed to be easy to be understood by lay people. So I use a lot of that covers here. This research is one of the first. This is actually funded by the first Komen grant to study why certain breast cancer early stage breast cancer are more likely to relapse versus others that are cured usually after surgery and adjuvant chemo. And so this is the paper that we discovered a gene called MET-adherent, MTDH, that have dual functions to promote metastasis and also chemo resistance. And these are the two main reasons that patients die from metastatic breast cancer. And that's why there's this double-headed snake as the illustration about how dangerous this gene could be. And later on, we have been working on this gene since then for the last 10 years. From discovery of gene to making genetic knockout of the gene in the mice to demonstrate that it's essential for different stages of breast cancer progression. And then a couple years ago, solve the crystal structure of the protein. And this is basically looking at the molecule at a blank, close distance to see how do you block that. And we see that because you can see this is the green protein called SND1 that work with adherent MTDH to cause metastasis. And the two areas in purple are the ones that adherent hold on to this protein to cause all the trouble. So if you ever go bowling, right, you use fingers to stuck into those holes to hold on to a bowling ball to bowl. And so that comes with the idea that if we you fill those holes with stuff into it, you cannot hold on to it. And maybe we could intercept the function of the protein. So this is exactly what Ming Hong, our latest Komen postdoctoral fellow, is doing in the lab. Is trying to discover a small molecular chemicals that stuck into those purple holes in the protein and to prevent that. So I'm happy to report that we already screened many compounds and we come up with a compound called C26 that do show therapeutic effect in the mouse. So hopefully this will be the next line of complete novel therapy for breast cancer. So a lot of work in my lab also focused on bone metastasis because many late-stage breast cancer patients suffer from it. And this is a discovery that was made many years ago with a MD PhD student in the lab. There you can see the photo of him. There he's now a fellow at Dana-Farber Cancer Institute in Boston now. So about 5 years ago, we discovered that there's a signaling pathway, a molecule called JAG1, that allow the tumor cell that you see in blue there to communicate with the bone cells and actually force the bone cells as forced labors for them because the tumor cell actually cannot break down the bone. The fractures that you see the patients are actually not caused by tumor directly. It's the tumors giving instructions to the bone degrading so-called osteoclasts. Those are the bone breaking cells, and make them hyperactive, use them to break down the bone. So this is what we figured out about 6 years ago. And in the last 5 years, we worked with the company that some of you heard about, Amgen. Amgen is the company that developed denosumab, the antibody that is used to treat bone metastasis right now. And you can see we developed this antibody, and we test that in the mice again. So you can see the left is the mouse that developed a lot of bone metastasis without any treatment. The second one is the one that is treated with standard chemotherapy. So it has some effect in reducing tumors, but tumors still grow quite a lot. The third one is the antibody alone. So you can see some therapeutic effect, but there are still some residual tumor. The most amazing thing is when we combine this new antibody with chemotherapy, it almost complete wipe out. You can see the last mouse is almost clear from the tumors. So this is a study that we recently submitted, and actually the review is very positive about it. I can share with you. As one of them said in the writing, you don't often see that in the review, saying that I hope this move on to clinical trial. And this is exactly what we hope to work with Amgen to move this into clinical trials in the patients with bone metastasis. The last one is the latest one that was just published last month. This is again done by a Komen fellow, supported by the Komen Fellowship, Tony. So Tony did this study to try to figure out why certain cancers always have recurrence and metastasis. And that is because they have the so-called cancer stem cells. These are the root of cancer. And the immune system naturally have a way to eliminate the cancer cells. But what Tony showed in this paper is that the cancer cell, that upper right one, this is in the artistic form. He's an artist. His father-in-law is an artist from Spain. So he drew this picture here to represent the tumors escaping from the immune cells, like macrophage, that produce interferons to have a suppressive effect in the cancer stem cell. But the cancer stem cell come up with a way to escape that. And this is what Tony figured out And by knowing that, maybe we'll come up with a new way to prevent the function of cancer stem cell. So the last one is the one I currently are supported by as a Komen scholar through the Komen leadership grant to study the dissemination of tumors to the bone to start the process of bone metastasis. And this is, again, a study showing that the tumor cells in green here attach to the blood vessels. And this is actually not a passive process. The tumor cells use a molecule called E-selectin ligands to bind to E-selectin in the surface of the tumor cell, and that is important to cause the tumor to grow as bone metastasis. So the good news, again, also is we work with a drug company, a biotech company called glycomimetics, because they have a compound that blocks the function of E-selectin. And we test that, again, in the mice. The top two are the mice without the treatment. The bottom ones are the mice treated with these glycomimetic chemical compounds. And you can see the bones is, there are less tumors in those color signals. Less tumors in the bottom two, and the bone is also protected. They do not develop as many bone metastases. So as you can see, what we try to do in the lab is to try to figure out what is the root cause of all these metastasis events in the patients. And by knowing so, we can then develop molecules to intercept them. And this is what we do in the lab, and this is what I am training my students to do when they develop their own lab as an independent scientist. So the last slide I want to show you is this is not just my own single personal effort now, because I was fortunate to work with many of the most talented students at Princeton University. So in the last 12 years also, about 11 postdocs have received training in my lab, 14 graduate students, and almost 30 undergraduate pre-med students. And most of them, 90% of them, went on to study medicine or MD, PhD. So those are the people who will be trained not only as a physician, surgeon, but also well-trained in science of breast cancer. And these are going to the patients who will be treating our patients with breast cancer. So these are some of our latest, graduate of the lab. You can see they move on to establish their own lab in China, in Beijing, Shanghai, at UPenn, University of Notre Dame, in Jackson Lab in Maine, and also in Europe. So this has become a global effort. We are metastasizing as well in a good way. And this is not one-time effort. By training this student, and they are going to train more and more students, we are going to grow our community of researchers and surgeons and people who develop drugs. Through this combined effort, we are going to defeat the cancer and achieve this common goal that we share with Komen to reduce the deaths of breast cancer by 50% by 2026. So with that, I want to thank you again for your support, because it's you who make all this work possible. Thank you.
On behalf of Dr. Kang, I want to thank all of you for joining today. I would also like to thank Odonate for their matching gift for all donations made to Dr. Kang's and the other 3 scientists shown here for his crowdfunding campaign. This generous gift allows for your donations to go even further. The link to the site is on the screen, which is bemorethanpink.org. I will leave the site along with the webinar open for a few minutes to allow viewers to submit any questions they may have for Dr. Kang to answer via email, and to note the website for the crowdfunding. Thank you all for joining.